WH reporting. Coral reefs are dying at an alarming rate as water temperatures rise worldwide as a result of global warming, pollution, and human activities. In the last 3 decades, half of Australia's Great Barrier Reef has lost its coral cover. A new study from Columbia University provides more evidence that genetic sequencing can reveal evolutionary differences in reef building corals that one day would help scientists identify which strain could adapt to warmer seas. The findings published in Science July 17 provide a window into the genetic process that allows some corals to resist dramatic climate shifts that could complement or improve current preservation efforts. We need to use as many tools as possible to intervene or we will continue to see coral reefs vanish, said Zachary Fuller, a postdoctoral researcher in biology at Columbia and first author on the study. Using genomics can help identify which corals have the capacity to live at higher temperatures and reveal genetic variants associated with climate resilience. Coral reefs found throughout the world in tropical oceans are one of the most diverse and valuable ecosystems on Earth. They are actually living animal colonies and important for many reasons. Reefs provide a habitat for a large variety of marine species, protect coastlines from storms, floods, and erosions, and help sustain the fishing and tourism industries. In the late 1990s, reefs worldwide experienced their first wave of mass bleaching, which occurs when high water temperatures destroy the symbiotic relationship with the colony's colorful algae, causing the corals to turn white. The loss effectively starves them, as corals are dependent on the photosynthetic algae that lives within their tissues for nutrients. Reefs can recover from bleaching, but prolonged periods of environmental stress can eventually kill them. The Columbia research predicts to some degree which corals are likely to withstand unusually high temperatures and resist bleaching events. Genomics allows us to examine the genetic differences that could influence survival and bleaching tolerance, helping us work out how we might support coral health, said Molly Presorki, professor in the departments of biological sciences and system biology at Columbia and senior study author. To collect their genetic data, Fuller and their collaborators from the Australian Institute of Marine Science analyzed 237 samples collected at 12 locations along the Great Barrier Reef, generating the highest quality sequences of any corals to date. The sequencing allowed the researchers to look across the genome for signatures where adaptation occurred and to find genetically distinct variations associated with the bleaching tolerance. What we discovered is that no single gene was responsible for difference in a coral's response to bleaching, but instead many genetic variants influence the trait. Fuller said, on their own, each has a very small effect, but when taken together, we can use all these variants to predict which corals may be able to survive in the face of hotter seas. Fuller said the findings after a pathway for coral biologists to further search for strains that can better cope with ocean warming and enables similar approaches that can be used in other species most at risk from climate change. The best chance we have to save what's left on the Earth's coral reefs is to mitigate the effects of climate change by rapidly reducing greenhouse gas emissions, Fuller said. In the meantime, genetic approaches may be able to busy us time.